Hello and good morning from Tel Aviv. And I have today a very unusual pleasure and honor to be with you with Professor Levenberg. And we are going to talk about the distinctive feature of tissue engineering. This is going to be probably the most fascinating conversation I had in a while. And Professor Shulamit Levenberg is from the Faculty of Biomedical Engineering of the Technion. She's the director both of the Technion Center for 3D Bioprinting and the Rina, Avner, Schneur, Schneur Center for Diabetics Research. And also she holds the Stanley and Sylvia Shirvan Chair in Cancer and Life Science in the Faculty of Biomedical Engineering. So good morning to you, Professor Levenberg. How are you, first of all? And I want to thank you, first of all, for all the wonderful things that you're doing for humanity. It's a great honor, and I'm sure you're going to, people are going to be not only so inspired by what you're doing, but probably going to finish, when we end the conversation, are going to be so much more hopeful regarding so many problems that we have to tackle, so much suffering that you're doing the best possible to end or to, you know, upheaval the suffering from us. So here we go. So how are you this morning?
I'm fine, thank you. Thank you very much.
Wonderful, wonderful. I was wondering if it's possible, if it's not too personal, to ask, how come you find yourself in this fascinating, you know, specific medical, you know, profession? Would you care to share with us a little bit about how your personal journey that led you here?
Yeah, so when I thought about what to go and study for my undergrad, I thought about different things, and I read about biomedical engineering, and I was very much interested in this. I actually mailed to the Technion and I asked them for material about biomedical engineering, but back then you couldn't study it for undergrad. It was only for a master and PhD. So I decided to start with biology. And then I went to the Hebrew University and really liked biology and started to work in labs there. And then for my PhD, I continued with biology. I went to the Weizmann Institute to study molecular biology of the cell and how cells interact with one another. And when I was thinking about a postdoc, I read about tissue engineering. And I really liked the idea about growing cells in 3D and really understanding how cells interact, something that is really mimicking better the tissue. So I decided to go to MIT, to Bob Langer Lab for tissue engineering. And actually I went to biomedical engineering because tissue engineering is really one of the core fields in biomedical engineering. And after the postdoc, I was recruited to the Technion Biomedical Engineering. And it was like closing a circle. And it was really exciting to find myself there. So
this is wonderful. And from the way you're speaking about it, it seems that you're, you know, getting up every morning extremely happy that this is what you're doing with your life. This is my take on the way you, I mean, you have so much passion talking about that. So this is what is being conveyed, that you're really happy with what you're doing. So
yes, I'm really happy, and I'm really glad to be at the Technion. Now we have undergraduate studies in biomedical engineering, and we have more than 100 students every year.
Wow.
Coming to learn this and to get professional in biomedical engineering, and many master students, PhD students. So this field is really, really growing very fast.
So what you're doing, what you're saying is, but besides your scientific work and this wonderful contribution, you're also assisting in creating the next generation of researchers and inventors and innovation and so forth, innovators and so forth.
Exactly. Yeah. Yes.
So how do you divide the day? I mean, between, you know, the lab and, you know, your scientific work and teaching. And I'm simply wondering, are you sleeping at all?
So I think most of the work is in research, but of course I'm also teaching. So also when I'm saying research is supervising the student, the PhD student in their research. So it's combination of the science and the teaching. So I think it's really very nice combination.
Wonderful, wonderful. So would you like to tell us a little about, I don't think people totally understand, these are new terms. People are used to, going to the doctor, the healthcare, the hospitals, but I don't think that many people really know what is, bioengineering or, bioprinting. And so would you care to, to teach us a little bit about what does it really mean?
So tissue engineering, the idea is to build tissues in the lab. So you grow them usually with a kind of scaffold that supports the cells, mimicking the extracellular matrix, so the cells can sit on it and proliferate and differentiate. So we're making pieces of tissues in the lab, and now when we have the new tool of 3D bioprinting, you can already decide how to put the cells in the right location and how to build the scaffold with the printing. But the concept is still to build a tissue that can be later used to repair, to replace damaged tissues in the body.
That's unbelievable. So how this method was created in the first place?
I think the concept was started by Bob Langer and some colleagues. Bob for many years worked with biomaterials, materials that can be degraded, and he mostly developed them for drug release, so they can really biodegrade slowly and the drug can be released. But then at some point it occurred to Bob and to some of his colleagues that these biomaterials can also be used to grow cells on them. And because they are biodegradable and biocompatible, they're okay to implant, you can seed cells on them and you can grow the tissue on them. And this is how this is one of the directions that how the field started, with this scaffold, with the biomaterials. That's
wonderful. So in a sense, you were part of the group of the pioneers, if you were in this lab and you saw this, how the profession is in a sense being created am I wrong?
Yes, I think so. And I was really very lucky to be accepted to Bob Langer Lab and to be part of this Langer network. Which is now spread all over the world because many of his students are in faculty position and in important jobs in the industry, and also founded companies in the field. So I think I was really lucky to be able to come. And actually I came from a biology background, and Bob is in chemical engineering at MIT, and the lab was very, very interdisciplinary. People came from different fields with different type of engineering, medicine, biology, science. And we all worked together, and this was one of the exciting experience that I really liked when I came back to the Technion in the biomedical engineering. You have the same concept that people are coming with different backgrounds, and the field is very interdisciplinary. You have to know different type of engineering and different type of science, and to combine it with medical studies to understand how you can build things and develop new things to improve medicine.
Wow, that's wonderful. So would you like to tell us a little bit about I know that you have various different projects that you are working on, and why won't we start with the one that is very dear to your heart and that you think that, it's really a new thing that is happening right now? Would you like to tell us about what right now you're working on and what is really dear to your heart? And
so I can start with a very recent project.
Yes, of course.
That was just published. And this is a research about engineering skeletal muscle tissue, a piece of muscle that can treat type 2 diabetes. And this is something very, I think, unique, because it's not a traditional way to think about how to reduce the blood glucose level. It's not by a different type of drug that reduce it. It's just a different concept. And the concept is to take the cells of the patient, the muscle cells, and just treat them and teach them how to do glucose uptake in a better way. So we take the cells and we genetically engineer them to have more of the transporter that absorb glucose from the blood. So we now have cells that can do it better, that can uptake glucose much faster. And we build a piece of muscle from these cells. And when we implanted this piece of muscle, they changed the whole metabolism of the diabetic animal, and the blood glucose level was reduced by the activity of this tissue engineered muscle. And actually we treated the diabetes. So this was really, really exciting. And we're now starting to move it, to think how we can move it toward the clinic.
So just to make sure that I understood, because I'm not I'm not a doctor. So what you're saying, you're re-engineering only a small part of the body in a sense, but then the impact is on all the body in a sense. Did I understand correctly?
Yes. So this small piece of muscle really affecting the other muscle. There are many ways of communication that the part of the tissue in the body can communicate with the whole body by secreting all kind of myokines, all kind of factors. So this piece of muscle, beside the fact that it can uptake glucose faster, it's also affecting the other muscle to act better also.
Meaning like the other muscles are learning from this. He's becoming the teacher. He's teaching them.
I guess so. You can look at this way.
I'm just metaphorically, but this is interesting. So how do you make a decision which muscle, or is it a specific muscle that you already know that? How did you make a decision regarding which muscle is it?
No, I don't think it really matter which muscle. And I don't think it really matter where you implant it, but in the muscle, but we implant it in the muscle. So, yeah.
And just to give, I think, our listener an idea, I don't know, but we're talking about millions of people that are suffering from diabetics worldwide. And I think one of the problems with diabetics type 2, that people don't even know when they got it. I mean, you can be pre-diabetic and you don't know it. And the last figures that I read, and unfortunately, it's also when we're thinking about health, we also have to think about the economics. So I think right now, only in the United States, they spend about $300 billion a year. It's going to get to $6 billion, like in 6 years. And by 2030, it's going to be $1 trillion only treating people with diabetics. And the suffering right now, there is such a horrible suffering. So this is a game changer, isn't it? If I'm not mistaken.
I hope so. If we will be able to use this technique to treat patients, this will be amazing. This will be really a game changer. We still have some steps to do before it can get to clinical trials, but I think it has a lot of potential. Yes.
So it had a lot of potential. You were treating what, rat, mice? What?
Now we work with mice, and we use different models of diabetes in mice. In 2 different models that we used, we saw the effect and we saw the cure. So now we have to move on toward working with human cells and proving that we can do it also.
So what the next step? The next step is human cell, and then human.
Yes.
Okay. No primates. I mean, you're not doing it on other. Start from the mice.
We have to see what the regulation asks us to do, and based on this, we'll have.
Wow. Is there a timeline for that? Just in general, so people can understand what does it mean. So
yeah, it's several years. It's years till it can reach the stage of.
And how many years have you been working on it till now?
It's about 5 years.
Wow.
Research.
Yeah. And how come?
To the point of this proof of concept that this can really be done. Yes.
But how did you even come with such an idea? Because all the pharmacy, all the industry is focused on medicine and insulin pumps. So how come you came with this? It's a total different definition of the problem, and total like the total different reframing the problem. How did you do it? I mean, how did you think about it?
So I think that a lot of credit should go to Rina and Avner Schneur. They really wanted to make a change in the research and hopefully also in the treatment of type 2 diabetes. And they came to my lab and asked, they said, we can donate money to fund the research, but we really ask if you can come up with a new idea, something which will be out of the box thinking and a new way to treat diabetes, not the conventional way. And I think due to this motivation, we really sat and had a brainstorming with some colleagues and came up with this idea of using my knowledge in tissue engineering and in muscle tissue engineering, which I've been working on it for many years, to try and use this to type 2. And because we have the funds, we could really start a new project really from the beginning, and also have be able to take the risk of a project which is really, you don't know if it will work or not. You have to, so actually it's always based on some other research. This is how research is done. You always based on other data. And we saw a paper where transgenic mice, where all their muscle was changed to increase this transporter, were resistant to diabetes situation. So we thought, okay, in humans, we cannot make, the patient transgenic, but we can maybe engineer a small piece of muscle, and maybe this can also have a systemic effect. And this is how we came to this.
May I ask, do you think this kind of, this solution, the concept, can it also be in the future a solution for Parkinson's disease? A problem that has an impact on all the body, like I'm talking about Parkinson or whatever. What do you think? Or LSA, I don't know. What do you think? Because right now it's more than diabetics. You're creating a different path of treatment.
I think that when you have a tissue that can be implanted and fully integrated, like we showed with the skeletal muscle tissue, that we can engineer it and it can be fully integrated. So when you have this in mind, I think this piece of muscle can be a vehicle to do many things, not only to improve glucose uptake, but it has a potential to do other things that are needed in the body. So I think it is a new concept that can be used. Not only injecting or cell, or like cell therapy where you come and you inject cells, you really build something and you integrate it fully with the body, and it's staying there and it can function to improve situation.
That's unbelievable. So let's assume that we right now 20 years into the future and we are living in fantasy world. Just for a second, in your imagination, what would you like to heal with this, new concept, new therapy? What would you see? Let's see if we can speed 20, 30 years into the future.
I think there will be many opportunities as we go on, but I think metabolic disease is the first, not only diabetes, but other metabolic disease. But I think other things as well.
Which metabolic disease you're referring to?
I think I don't want to now to mention.
No, of course. No. I just was wondering.
Still in the, working on the concept and working on.
No. I'm sorry.
Early to really mention them, but.
No, it's okay. No, I didn't. I'm sorry. I was so fascinated because I said, well, perhaps we can cure anything with this. Perhaps I'm too ambitious, or, perhaps I'm not realistic or too optimistic, but because what you're really saying is, in a sense, you're creating a method that will assist the body to heal itself. This is.
Yes, I think, yeah. And I think this is part of the great idea of tissue engineering, when you use a real tissue to repair or replace damaged body. And now we see that it can also be repairing not only a specific tissue that is damaged, but also to heal something more systemic.
That's unbelievable. How did you feel when you said that it was, at least it was succeed, you saw the success in with the device? How the team and you, how did you feel?
It is very, very exciting when you think about something that is, you're not sure that it will work. And then, but you say, there is a lot of chance, and that, and you do the search, and you know that the methodology makes sense and it should be okay, but still, till you do the experiment, you don't know for sure. And
no, of course,
it's really very, very exciting, and it's a lot of work. And Rita Beckerman, she's a PhD student in my lab who really did the project and led it in the lab, and it was, a long journey, and she was really very stubborn and worked very hard, even though it was a very long journey. But I think at the end, when you get to the point where the research is completed and you submit a paper and it's getting accepted and you see that the results make sense and it's had the potential to help patients in the future, it's really bring you a lot of satisfaction. And I'm sure she's very happy about the point that she reached.
Yeah, amazing, amazing. I think we are all gonna, look for, and we are going to look forward to hear what is going to happen further with this, because it's really it's mind blowing. I'm sorry to say this in a such not a nice way, but I think it's a game changer. It can be a game changer. It's unbelievable.
And I think that part of the thing was that immediately right at the beginning we interacted with Professor Eddie Carnielli, who was the head of endocrinology in Rambam. So he's a physician. He worked with diabetic patient, and he also happened to work with the transporter, with the GLUT4 that we decided to focus on. So I think this collaboration between clinicians and a researcher is also very important when you think about clinically relevant problem.
That's wonderful. That's wonderful. So this is probably takes so much of your time and imagination, but I know that you have few other things that you're still working on, or would you like to share what is the next one?
I can, if we're talking about interaction or collaboration with the clinical world. So I would like also to mention our recent work, something that was also very recently published, the work on the ear reconstruction, bioprinting of a ear. And this project started by a clinician, Dr. Duvdevanik from Sheba Medical Center, that approached me and said he's working with kids that are born with a ear that is not normal, something that is necrotic ear. And
what is necrotic ear? I'm not sure that.
In some condition, the child can be born with one ear that is normal and one ear that is very deformed. Can be very small or very deformed. Sometimes both ears, but usually it's one. So and then they come to him to try and fix it. And the method that he's using now is very, very painful for the children. And he's heard about our research in tissue engineering and about our 3D bioprinting approach. And he said, maybe we can do something together. Maybe we can think about a way to 3D bioprint the ear, that it will be the same as the other ear, and without taking a cartilage from other place in the body that is really, it's very painful. So we decided to take this challenge, and the student who worked on it was actually a graduate student in my lab, and she stayed for a postdoc, for a first postdoc in the lab, Dr. Shira Landau. And we said, okay, we'll try and find a method to bioprint the scaffold, the material that give us the shape of the ear with all the details and how it should stay and remain in the right shape and not being like damaged after implantation. So we designed a way to bioprint the scaffold and seeded cartilage cells on it, and really made cartilage on the outside the body in the lab that later could be implanted and keep the shape of the ear after implantation.
So, meaning you're starting ex vivo, meaning you're creating the ear outside, and then how do you implant it? I mean,
so we implant it under the skin, so the skin covered it, and
so it's a surgical procedure, but it's already a hole here is being created, meaning he's getting a whole. Wow, and it's probably adapted to his size and his age and so forth. And
yes, you can do a CT scan of the other ear, and then we build the printing exactly based on this specific shape. So you print it according to fit the other ear exactly.
And there is the question of the functionality of the ear. I mean, is there a difference in the ability to hear or the level of, I mean, the hearing is impeccable, the hearing of the new ear. I mean, it's totally functional.
Usually it's not supposed to be fix the hearing. It's only like external aesthetic. But sometime during this surgery, some physician can also open the canal. So if the problem is there, then it can be open and fixed. But most of this approach is aesthetic, just the outer part of the ear.
Well, but of course, because a child, if he's, you know, he looks very different than this. So this is heart-shattering to think about all these small children only born and already, you know, so where are you in this process? Where are you right now? Are you still in the lab? Are you outside of the lab? I mean,
so we showed the concept and we published the paper showing the methodology, how we can do it. We also showed that we can implant it and the ear can keep its shape. And now we are trying to move it more toward Sheba Medical Center. So they build the lab there. Based on our instruction, they build a tissue engineering lab there so they can continue with the research, and hopefully from there, they will be closer to bring it to the clinic and do it for kids. But there is some research to do and some approval that need to get to be able to do the clinical trial.
And here, what is the timeline in your opinion in general? What are talking? 5 years, 10 years, 15 years, how long?
I don't know how to say years. Also, in all of these projects, when you want to reach the stage of a regulatory pathway and safety test, then it's also you need a lot of funds. So it's also depending if you're able to raise funds. So usually, and you can start a company, or a company can decide to license the technology and invest some money in it, so to be able to move it all the way toward the clinic. So there are some steps that need to be done that are outside the lab.
No, of course. But this is, and how many children worldwide, in your opinion, are suffering from this problem?
I don't remember the numbers.
Okay. But unfortunately, it's more than 10 a year or something. Otherwise,
And also, it's not only for kids. It's also for patients that there is a traumatic injury, and the ear need to be repaired or replaced. So there are many other conditions where you need this reconstructive surgery beside the kids that are born like this.
Do you think
car accident or other?
Yeah, that's what I thought. So you can use this method to go beyond the ear. It can be the whole face, no? I mean, people are, unfortunately, you know, we have here the army and people might be burned, or so can this method go beyond and, you know, reconstructing the whole face if it's needed?
So actually we have a big grant from the European Commission for a facial reconstruction, where we try to do other pieces of tissue and also to do multi-layered tissue when you need not only this cartilage, but sometimes you need the bone, the muscle, the fat, like all the layers together, the bone to be replaced. So we're working on this direction. And the critical step is how you can really vascularize this tissue, how you can bring blood supply to the tissue, because this will allow you to go into larger tissue if you're able to feed the tissue outside the body and also connect it right to the blood supply after implantation. So we are focusing on this direction, how you can create the blood vessel network in a tissue when you engineer a tissue.
So this is a big hurdle that you have to go. You come over in order to be successful in all the other domains. It's like, you know, it's like switching on and off the light in a sense in the whole system.
Yes.
I'm sorry that I'm taking such, you know, horrible metaphor. I'm just trying to simplify it for people who are not from from the field.
Yes,
wow, yes. That's wonderful. And is the doctor that approached you are happy with what you're putting on the pediatrician that? Yeah.
Dr. Devani from Sheba, yes, I think he sees a lot of hope in this technology and in this direction, and we're continuing to work on it. We actually got funds from the Innovation Authority to continue and do this translational part of the research to try and move it from the lab toward the clinic. So we continue with the research and hoping to be able to bring it to patients.
So this is a wonderful collaboration between, you know, the science and what you're saying, the real, I mean, the people who take, I mean, the doctors, you know, in the medical field, and this is a wonderful. Are there any more collaborations like this that people approach you and say, well, we have this problem and you think we can solve it somehow together?
Yes, it's happening all the time.
Oh, that's wonderful, but that's wonderful.
I think another very important example is our work on spinal cord regeneration. And we were approached by the Israel Foundation for Spinal Cord Injury. And it was some years ago. And they came to us and said, if maybe we can use our tissue engineering techniques to repair spinal cord injuries. So people that are paralyzed can come and start to walk again. And at the beginning, you know, I said, okay, I have to think about it because I was not working in this direction before. We were working on muscle and on heart muscle, on pancreatic cells and other, but not on spinal cord. But we decided to start a project on it. And it started with a project of a scaffold that is seated with stem cell and implanted in the injury site. And we saw a lot of success. The rats started to walk again after implantation. Wow. And this was really amazing. After really that they were fully paralyzed, they started to walk again. But then we decided that maybe this will be a little bit difficult to translate into the clinic, this implantation into the injury site. And we thought about a new direction. And this was also with the fund from the Israel Foundation for Spinal Cord Injury, and also from a fund that we got from Shirvan, a donation for research and a research fund. And we decided to go and use small particles that the cells secrete and use them to treat the spinal cord injury by modifying these particles. We call them exosomes. These are vesicles that the cells secrete. And we decided to modify them and put in them siRNA that inhibit a molecule that inhibiting the regeneration. So when there is an injury, there is a molecule that is going up, a protein that is elevated and somehow inhibiting the regeneration and the axon ability to elongate. So we decided, okay, if we will inhibit this, we will be able to allow better regeneration. The axons can elongate and reconnect, and then we can get the repair. And this is what we decided to do, and this really worked. We gave the small particles intranasally, so they can reach the area of the injury and concentrate there. And again, the rat started to walk again. Wow! So this was really amazing. So this was mostly a work of Dr. Shaway, Joe, in my lab. Wow! And this was a collaboration with the lab of Professor Danny Offen from yes. Tel Aviv University. Yes. And this is really very exciting because we founded a company, Nurixon, based on this technology. And the company will work on how to bring it to the clinic all the way. Again, it's a long journey. But this is something that is really non-invasive. You can give it through the nose or by injection. And if it can bring people back, to normal, back to walk again, or even just improve their paralysis in some sense. So this is really important.
This is extremely important because, once they are, we had a podcast here with a wonderful person that unfortunately, he's now paralyzed, and he talked about all the difficulties. People don't understand that it's while you're sitting all day, you're getting sick. I mean, suddenly your body is weakening and weakening. So you're becoming like a hub of difficult problems, just the fact that you can't walk. It's beyond the fact that you're losing your freedom and so forth. And also he said something very interesting, is that once people are in the wheelchair, they are becoming transparent. And he said that when he's sitting with his wife and he's a well-known scientist and he's a very serious, but the waiter is only speaking to his wife. What he would like he's an invalid, or an infant. What should we bring him? So but this is also, it doesn't feel like a miracle, meaning you're giving a shot or you're giving something through the nose and then suddenly the rat's starting to walk. I mean, what does it tell us about the ability of the body if we do it correctly to repair himself? I think
it's amazing. I think, the body has a lot of power to regenerate, but sometimes we have to help the body to find the way to do this regeneration properly.
So, no, that's what I'm thinking, but I mean, it's just, I'm not a religious person, but you think about this beautiful machine that has so many abilities. We don't even think about, how well it is taking care of us. We're breathing all the time without even thinking, right? The air is, it's no, and then what you're saying is that how can you, assist and push the body to recover? It's a kind of a very interesting collaboration, right? I mean, it seems nearly, it's beyond, in a sense, even though it's science, but in a sense there is a sense of magic. And I'm not a spiritual person as well, but I'm saying, isn't it? Don't you feel that it's unbelievable, right?
Yeah, I think it's amazing. It's very, and we know so little. And whenever you learn more and more, you just understand how little you know from the big picture. But It's still amazing.
Wow. Wonderful. So right now, and it's good. It's wonderful that again there is a collaboration and also there is a funding. So, meaning people understand the importance of it, even though there are going to be many fails on the way. They understand that it's a road that's important to take. It's a journey that is worth taking, even though it's a long one, and especially when you start with something that is new, a new way of treatment, a new concept, then it's a longer journey. But I think it's very important to do it. But it seems to me that you're always looking for a new and innovative way. This is my take on the way you look at because the traditional ways are not good enough. So it's not that you diminish them, but there must be something better.
You always build on previous work.
Yeah, of course,
you always rely on other people's work.
Yeah. We are all sitting on the soldiers of giants. So, but on the way you talked about something. On the way you talked about heart muscle tissue and pancreas. Do you want and bone? And you said it on the way as if it's not. Would you like to talk about this a little bit? I think it's very interesting as well. If it's okay that I'm asking, of course.
Yeah. So the concept, the thing I think that tie all this tissue is the fact that we really wanted to show that we can increase penetration of blood vessels into an implant, because let's say we engineer a piece of tissue and then we implant it. The body need to nurture it. The body need to send blood vessels to bring oxygen and nutrient to the piece of tissue that we just made. And we found that if we add endothelial cells, the cell that really line the blood vessels, if we add endothelial cells to this mixture, they can spontaneously form the tubes and spontaneously form the network. And we demonstrated it both in skeletal muscle tissue and in cardiac tissue, as a patch to the heart, and also with pancreatic islets, that we can get pancreatic islets. And if we seed them with endothelial cells that form all this network, they can function better and really survive better in the lab, but also after implantation. All this network that was formed in the lab, this engineered vessel network, can secrete signals that bring the host vessels faster into the implant. And also what was very interesting to see that we saw connections between the engineered vessels and the host vessels.
Wow.
Anastomosis, and blood can now start to flow from the host directly like a highway into the inner part of the implant. And this really facilitates the perfusion of an implant and also its survival and function. So this is really, I think it's a very important step and a very important knowledge in tissue engineering. And not only my lab is working on it, but other labs around the world realize the importance of this co-culture, adding other cells, not only muscle cells, but also adding these endothelial cells and other cells that can help to form the tissue in a better way that mimic better the real tissue. So, but these pieces of tissue, we first demonstrated them as an implant. So you have the piece, you implant it, and you wait till the host can penetrate and feed the implant. But in the clinic, when there are larger damages or larger pieces that need to be implanted, the clinician usually will use tissue flaps, meaning that they come with their own blood supply. They will take a piece of muscle or from one part of the body, but they will take it with this artery, with the vein, with the large vessels, and do microsurgery to connect it to the larger vessels in the right place. And by this, when the blood flow, immediately everything is fully perfused because you transfer the tissue with its own blood supply. So we decided to make this concept, do it also in tissue engineering, to engineer tissue flaps, meaning that we can now have them engineer not only the small capillary network, but also engineer the large vessels, and we can perfuse through the large vessels, and also later on connect them. When we do the implantation, we can connect it to the large vessels. So in this concept of making tissue engineered flap, we decided to focus also on bone, be able to make a bone tissue flap. So Idan Redinsky in my lab worked on bone tissue flap, and this was a lot of work, but this was really very exciting to see that we were able to engineer a bone tissue flap, that we can take it now to repair bone defect, but bring it with its own blood supply. So the regeneration, the repair of the bone can be much, much better. And also we decided to take it another step and use 3D bioprinting to make this tissue flap. And Ariel Shaklani in my lab, he already graduated, and he was able to show that we can engineer large vessels and they can connect to the smaller capillaries, and we can engineer muscle tissue or cardiac tissue around it and implant it. And this can now connect to the blood supply immediately right after implantation, and the tissue can survive. So this is a way now to go and to engineer larger pieces of tissue. When you have the way to make this whole network, multi-scale vascular.
Wow. So where is this project right now? I mean, is it in the lab? Is it already being used in the clinician? I mean, where?
So it's still in the lab. We still have a work to do. We still need to enlarge the tissue to make it larger. And for this, we're using also 3D bioprinting to make larger tissue. When now you have, the design is more complex than the smaller tissues. So we're working now on this to make the tissue larger and still be fully perfused and get its own blood supply. And in the lab and also connected after implantation.
Wow. And looking into the future, let's assume that it's going to go out of the lab and into the clinics. What do you think, what in your opinion is going to be used for? What is going to replace, or is it new treatment that never, wasn't available? How do you think it's going to be used in the future?
I think if we will be able to engineer larger pieces and then implant them, so we can think about many ways that it will be used. Let's say you have a large piece of missing of bone and muscle and you want to implant it. It can be after injury, or it can be following, let's say, a tumor removal when you have an area that needs to be now replaced with a new tissue. So if you don't, so sometime now you take the tissue from another place in the body to fix the damaged area, but now you have two places that are being damaged. But if you can engineer it in the lab and use it to repair, then I think it's much better.
Just to make sure that I got it. For example, if unfortunately someone had cancer and an organ had to be removed, we might be able to replace it with a new tissue, even with this, the same genetic code, and to let it grow inside the body in a sense?
Yes, exactly. Yeah, this is it.
Meaning science fiction at its best, or for example, instead of doing a full heart implantation, I mean, really, taking another heart and, replacing, do you think this can be a way to, tackling the problem where the part of the heart that is usually being hurt after the first cardiovascular event? So it might be also a way to repair the heart without doing a full transplant. So if I'm asking stupid question, it's okay. Tell me. I'm good at asking stupid questions. So I'm just.
I think right now we're more in the tissue level. Organ level is more complicated, because with organ you also need, it's not only to repair or replace a piece of damaged tissue, you need the organ functionality. So this is another level, but around the world, people are working on how to get to this stage. So we are also, but it needs a lot of work to get to the point where the whole organ can function. But it's on the way. I think it's not there yet. We still have research to do to be able to engineer such complex organs, but
as you said, if someone had to, you had to move a tissue, now we can replace it, and I think it's wonderful. I'm just thinking about all these women who had the BRCA, mutation, and that cut, parts of their body because they want to make sure that they can be here and live for the children and be a part of their life for a long time, and perhaps now we can assist them with.
Yeah. We have a project in the lab on breast reconstruction.
Really?
Well. Using 3D bioprinting and scaffolds and cells to find a better way to make these implants. So again, it's in the research stage, in early research stage, at least in our lab. There are things that we need to develop. But I think it's something that we would like to be able to do to help patients like this.
That's wonderful. That's wonderful. I'm sure that if there is a heaven, you already have I think a whole skyrocket for you and your team. No, I'm saying you're really, it's taking medicine and science, and I think technology really and science is for assisting us, the human being. So I think it's beyond any imagination. Like, I think 100 years ago, no one would have thought that this will be possible, right? I mean, it's something that is started to being possible in the last 20, 30 years, even to dream about it.
Yes. And I think I'm also very lucky to be at the Biomedical Engineering department with really excellent students that have engineering background, but they're all their motivation to improve medical situation and to improve medicine. So this combination of engineering toward medicine, or engineering better life, as we say, I think this is the fact that I'm surrounded by such a bright student. This is something that's allow us to do the work. Without this,
there is a mutual inspiration circle. It's like a positive circle, so that everybody, the inspiration is coming from all sides, and so it's in collaboration. I think you have other issues that you're working on, and I don't think that our listeners believe anymore that you're sleeping. I'm just sure about that. But I'm joking, because you're doing such magnificent, unbelievable work. So anything else you want to talk about? I think you talked about, when we talked about lymphatic networks, bioprinting, when we talked about, do you want to?
About what?
lymphatic networks.
The lymphatic, right. Another project is...
I'm a good student. I'm learning everything here.
Yes, right. So we were able to show that we can engineer blood vessels and engineer the network. And then we wanted to see if we can also engineer the lymphatic network, which is the lympha. It's the different network than the blood. So different type of tubes, but still connected with the blood vessels, and the endothelial cells also build the lymphatic. So it's a little bit different endothelial cells, the lymphatic endothelial cells, but they're very similar to the blood vessel endothelial cells. And we decided to see if we can engineer this in the lab. And Dr. Shira Landau from my lab took this project on herself, and we mixed the endothelial cells, both lymphatic and blood, and we're able to show that we can build the network, the vessel network, the blood vessel network, and the lymphatic. And the lymphatic were functioning as lymphatic vessels, that they absorb fluid from the tissue, from the surrounding. And we were also able to implant this piece of muscle, skeletal muscle, with the lymphatic vessels, and show that they connect to the lymphatic vessels in the body. So you have the engineered lymphatic vessels connected to the lymphatic vessel. And we hope that we hope to continue with the research, and hopefully it can shed some light on lymphedema, and maybe also treat lymphedema by implanting now more active lymphatic vessels. Maybe they can function in absorbing fluid and helping lymphedema. So,
I'm not sure everybody knows what is lymphedema. Can you
when lymphatic vessels are not functioning, and then you see swelling of the tissue.
Ah, okay.
Yeah. So yes. So
wonderful.
We hope to continue with the research, and when we're talking about this vessel, it's also bringing another project that we are working on, which is how you can mechanically affect the alignment of the vessels. And we were able to show that if we stretch the tissue, if we do some mechanical stimulation to the tissue, this can affect the alignment of the vessels, the blood vessels, and also the lymphatic vessels.
Wow.
And this is very important because for some of the tissues, you have nice alignment of the tubes. So for example, in muscle tissue, you have the fibers and you have the vessel aligned in between the muscle. And the fact that by stretching, we're able to get such alignment gave us a better tissue for implantation. And we actually proved that if you want to implant into the muscle and the vessels are aligned, then you can get better integration and better perfusion of the tissue because they can connect better to the host vessels from the sides.
Wow.
So.
Wow. Wow, it's unbelievable. Unbelievable. We are nearly finishing. I know you were extremely kind with us with your time. Anything that you want to share that, which is only, in your thoughts, in your dreams, any other project that you want to talk about, any request? I mean, I understand, perhaps you're looking for some funding. I'm sure that because what you're doing is so important. So I think it's also worthwhile. I mean, whatever you want to share with us. And
I'm actually happy to share two other projects.
Wonderful.
One is
wonderful. No rush. I have time. I'm simply respectful of your time. I have enough time, so for as long as you want.
So a project that came out of the lab, and we founded companies based on this technology. One is NanoSynex, a company that is a small microfluidic device that was developed in the lab. This was a project of Yoni Shemesh and then Jonathan Avisar from my lab, a PhD student that developed this device that with very small wells, nanoliter wells, that can be used as a small chip to do early diagnosis. And we decided to focus on antibiotic susceptibility test. So you can test which antibiotics is relevant for specific infection, for specific bacteria. So we developed this technology, but then it was licensed to NanoSynex. We founded the company, and now the company is trying to push it to the clinics to have a much faster way to know which antibiotics you need to take when you're hospitalized or just when you have infection. And shortening the time is really very critical. And by this small device, we were able to show that indeed you can shorten the time. So
wow,
one technology that I'm very proud of. And
also it's you know, because it's stopping this process of, you know, trial and error. You do it, you simply erase it.
Yeah, and instead of giving just broad antibiotics, you can very early know exactly which antibiotics you need to take.
So it's a part of precision medicine in a sense.
Yes.
Personalized precision medicine.
Yes,
because
yes.
Wow. And this is going to be, I mean, in the end of the day, it's going to be like on every doctor, you know, clinic. Then instead of, you know, writing down the prescription for everything, well, let me check, and then this is the right one for you. So also it's going to assist with the waste and of course the suffering of trying and retrying and all of this. Wow.
Yeah, long hospitalization till you find the right antibiotics. So
wow.
So hopefully. And just to finish with
no, it's okay. I'm with you. I'm not in a rush. Yeah.
When we're talking about skeletal muscle tissue, we at some point one of my students, Tom Ben-Arieh, asked if we can try to use this technique of cells and tissue to engineer small piece of muscle that can be for food, to take instead of human cells, to take the bovine cells and edible scaffold and to engineer muscle tissue that can be used later as a steak for food. And based on this technology, we founded Aleph Farms that is not working on making these pieces of thin cut steaks for food. So it's a different direction than the medical direction, but not that important.
No, I think it's a different kind of medicine, you know, because I think the Rambam said, and you know, all other whatever you put in your mouth, it's also a kind of a medicine, right? You know, you are who you eat and what you eat and so forth. So I think it's not within the body, but you know, for external to the body. That's wonderful. That's wonderful. And it seems that also you're working with wonderful people, and there is a sense of I think mutual vision and mission and you know, collaboration, which is towards, you know, really improving human health and life and ending suffering as much as possible.
Yeah, we hope so. Yes.
Wonderful.
Thank you very much.
I want to thank you on behalf of all our listeners. For me, you are a source of inspiration, and you don't see it because it's the Zoom, but I have tears in my eyes a few times during this conversation, and I think I'm very blessed that I was capable of starting my morning with you. Thank you for everything that you're doing for us.
Thank you very much.
Thank you.